In this video we will discuss about circulating tumor cell interactions. So how the circulating tumor cells in the blood interact uh, that helps in their survival. We will start with this platelet interactions with the tumor cells. Platelets has this selectins, P-selectins, and also this integrin that can interact with the tumor cells that has their respective selective ligands, tumor selectin ligands that interacts with the platelets, and this helps in the aggregates formations of the tumor cells. And these aggregates, when tumor cells when present in aggregates, there is more probability of its survival. And the second interactions is about this neutrophil, where the neutrophil also helps in the aggregates formations of the tumor cells. However, how this neutrophil interacts with the tumor cells is not yet known. Some literature says that these interactions could be because of L-selectin or because of integrin. However, there is no consensus on the interactions of the neutrophil and the tumor cells. But when the neutrophils interact with the tumor cells, it also helps in the aggregates formations, just like or similar to the way the platelets help. The third way that tumor cells interacts, third is through to the endothelium cells, is via this P-selectin and also this E-selectin. And this time the tumor cells, this interactions helps in the adhesions adhesions of the tumor cells. And apart and this, what it does is P-selectin and E-selectin slows down the movement of the tumor cells, circulating tumor cells, and the Vcam and the integrin ligands, Vcams and the integrins ligands thereby helps in this uh, adhesions, firm adhesions of the tumor cells to the endothelium cells. And apart from these endothelium and tumor cells interactions, which is dependent on P-selectin and E-selectin, there is another way by which these tumor cells interact with the endothelium cells, which is not P-selectin and E-selectin dependent. That is through monocytes. So the monocytes can interact with the tumor cells, and thereby this monocytes helps in the adhesions to the activated endothelium cells through this L-selectin and the tumor selectin ligands that are present in the tumor cells. And this is also been supported by the chemokines that are released from the tumor cells, which later helps the tumor cells towards extravasations. So thereby exiting the tumor cells from the circulation bloodstream. So these are some of the main ways, some of the major ways by which these tumor cells, circulating tumor cells interacts, uh, that helps in the tumor metastasis and migrations. And so here some of you might be wondered why this the cells which belongs to the immune cells categories helps in the tumor cells proliferations. And this could be one of the reasons could be that these tumor cells are not been detected by the immune systems at this point of time to be considered as a foreign particles. And this is this could be one of the reasons by which at this point of time they are not triggering the immune response. As we know that there are two kind of response: innate immune response and adaptive immune response. So the adaptive immune response might take time. They might take time when the antigen presentation information is presented by the dendritic cells, thereby dealing antibodies against the specific epitopes. Till that time, the innate immune systems plays a role, and here the mature monocytes need to be present so that it can trigger the phagocytosis. However, Here the monocytes that are shown here does not seems to be mature enough to trigger the immune response. This could be some of the reasons by why circulating tumor cells are able to evade the immune system. At the same time, the tumor cells also have immune evasions capabilities such as high glycocalyx that prevents the tumor epitope to be interacted with the immune cells. That could also contributes towards this tumor cells immune evasion. So some of the things are not mentioned in this paper. This is based on my own knowledge. So if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.